In this video, we are going to finish up the topics of chapter 5. We are first going to be going over section 5.4, which has to do with protein analysis, but more specifically, how do we look at the amino acid composition of a protein? So understand that in the techniques that we will be discussing that look at amino acid composition, we're going to be studying the primary structure, of a protein. The first technique that we are going to see is Edman degradation. So at this point, you're just trying to verify that the protein of interest has the correct sequence of amino acid. And Edman degradation specifically is going to do so in the following manner. Edman degradation is actually a sequencing technique. You will be looking at the sequences of amino acid one at a time. But understand that in essence, how it works is that it's going to be taking the amino acids from the N terminus. That's why it's N-terminal sequencing. So if you have a particular amino acid, you will be studying one amino acid at a time from the amino terminus, going on and on and on down the peptide towards the C terminus. Overall, understand that the process of Edman degradation, it is complex. There are different chemical reactions that have to happen. We need to separate the results of those chemical reactions by high performance liquid chromatography, which is a technique that wasn't introduced. So understand that the logistic of talking about this is that the technique analyzes amino acid sequences from the N terminus one at a time. Now, there are other ways that we can also look at the sequence of amino acids in the protein. The second technique that can be utilized to look at amino acid composition is by utilizing enzymes that we know are going to hydrolyze specific peptide bonds. The first one that we're going to mention is trypsin. So, a cleavage with trypsin, pretty much what is going to happen is that we're going to cleave, in other words, cut peptide bonds after the amino acids arginine and lysine. So tryptine is a protease, which means that it actually hydrolyzes, the amide bond after arginine and lysine. So as you can see here in the example that we have alanine, arginine, serine, lysine, and tryptophan. So if we take into account that this peptide is going to be treated with trypsin, I'm just going to write it in a little corner of the right side of the slide. If we have alanine, arginine, serine, lysine, and tryptophan, if we treat this with trypsin, what we're doing is that we are hydrolyzing specifically after arginine and after lysine. So if we had this pentapeptide, because it has 5 amino acids, be cut by trypsin, we will have a little peptide that is alanine and arginine. We will have another little peptide that is serine and lysine, and then we will have by itself tryptophan. So we're only creating 2 peptides and then a single amino acid. Another enzymatic reaction that can be done for the analysis of the amino acid composition of a protein is chmotrypsin. Now, chmotrypsin is different from trypsin because it cuts the peptide bond after the aromatic amino acids. And as you can see here, your aromatic amino acids are going to be tyrosine, tryptophan, and phenylalanine. So as you can see, if the original segment of the protein that we see is the one given in the figure, after chymotrypsin, we have this peptide, which is here. We have this peptide, which we can see here. We have this peptide here. And then let's say that the serine is the last part of the whole system, then we have serine by itself. So in this case, chymotrypsin is creating 1, 2, 3 peptides in a single amino acid. There's one more type of cleavage technique, but this is through chemical means. So cyanogen bromide, it is a chemical tool that can be utilized for cleaving peptide bonds. Remember, all of these are just going to be allowing us to look at amino acid composition. So overall, understand that specifically cyanogen bromide is going to cleave the peptide bond after methionine. Now, before we saw that the reaction was hydrolysis, we were converting an amide to a carboxylic acid at that end, and then the other end will have an amino group. In cyanogen bromide system, it is a different type of cleavage, because overall what you see here is that we have our side chain for methionine, which is here. That is going to be reacting with the cyanogen bromide. And the way that the cleavage happens is that overall, as you can see here, the bond cuts here. And what we have overall is that we are going to form a homoserine lactone with the C terminal. So in this case, it is not hydrolysis. It's going to be the formation of a lactone, because we're not utilizing water to cut. Overall, we're going to have our side chain for the methionine react with the cyanogen bromide to create this homoserine lactone. The last methods that we are going to be discussing in this chapter is specifically for looking at protein structure. The first one that we're going to talk about is Xray crystallography, followed by talking about NMR spectroscopy. When it comes to Xray crystallography, understand that in Xray crystallography, the main thing is that you need to have a protein that is pure, and you need a crystal. For Xray crystallography, we need a crystal. Okay? Understand that this is a very powerful method, because the majority of the protein structures that we know to date has been obtained through this. Now, once we have that crystal, understand that pretty much in Xray crystallography, we're going to measure the electron density, okay, of that crystal on extreme high resolution. So as you can see, the X-ray diffraction is going to give us a diffraction pattern of that crystal. And after you have molecular replacement, experimental phasing, which these are just the steps that happen, at least at the computational level, you are going to be repeating the step until you have the final model of the structure. The main thing that I want you to get out of the techniques is that in X-ray crystallography, we need a crystal. And in this technique, we're going to measure the electron density on high resolution. Now, the structure of a protein can also be obtained through looking at NMR. NMR stands for nuclear magnetic resonance spectroscopy. Understand that different from what we saw in X-ray diffraction, for NMR, our proteins need to be in solution. That means that they need to be soluble. So we need it in some medium. Okay? Now, understand that this method is going to be independent from crystallography. The main thing about NMR is that it actually measures the distance between the hydrogen atoms in the molecule. To just give you an example of the data that you obtain, you can see on the left side of the slide this little diagram that actually shows you the NMR data specifically for the protein lactalbumin. But understand that in order to to understand the overall structure of the molecule, you need to combine NMR with X-ray diffraction if it is available. But again, when it comes to what do I need to know about the technique is that the protein needs to be in solution, and what is we're measuring in NMR is going to be the distance between the hydrogen atoms in the molecule.